All right, in this video, we're going to be covering a pretty complex topic, action potentials and how they're generated and spread down a neuron. So if you haven't seen it yet, make sure you watch the video before this in the playlist that's covering resting membrane potential. And the quick summary is, in that video, we talked about how ions get transported across membranes in a neuron to set up resting membrane potential, which really just means that we've got a charge difference across these neurons. So as a quick recap, the way this happens is there's a sodium-potassium ATPase, which is just an active transport pump that moves sodium to the exterior of a cell, right, extracellular space, and moves potassium to the interior of a cell in the cytoplasm. And when you combine that with the fact that there are leak channels that allow potassium to slowly and constantly leak out of cells, so it loses some of that positive charge, the end result is that you get a cytosol that's a bit negative, and you get an extracellular fluid that's a bit positive. That really is the key to understanding resting membrane potential. Remember, the potential part of it is just telling you that there's the potential for ions to diffuse across the membrane, right? And since sodium is built up on the exterior, if we open channels that allow sodium to move, it's going to enter the cell, right? This is showing you a pump, but if we added just a regular diffusion channel and we opened it, sodium would want to go this direction, right, from a high to low gradient. Potassium would want to diffuse this direction because its gradient points the other way, high to low. And so the next concept in generating an action potential is deciding when we're going to let those ions move in or out of the cell, okay? And the first thing to pick apart is we have to talk about action potentials as opposed to something called graded potentials. And this graph is giving us a summary of what's happening, right? So we're starting off down here. This is our resting membrane potential. It tends to be somewhere around -70. And all that really means is if the neuron has no stimulation, nothing's happening, it's just sitting there waiting, it tends to camp out right around -70. If you want the resting membrane potential of that neuron to either go up or down, we're going to have to send some signals, right? And these different signals that you're seeing that we call either EPSPs or IPSPs, this stands for excitatory postsynaptic potential. Right, postsynaptic because in this neuron, this little green guy or these pink ones over here, these are representing the axon terminals of another neuron that's somewhere off the screen that we can't see. And they're going to be releasing neurotransmitter, right, onto their synapse. And so when we say an excitatory postsynaptic potential, remember, this is the postsynaptic neuron, right? This guy would be the presynaptic neuron. And when we release an excitatory neurotransmitter, it just means that it stimulates the next cell, right? So excitatory postsynaptic potential really just means we released a neurotransmitter up here at a synapse, and it makes us go up on this graph, right? Remember, anytime you're increasing your membrane potential, we call that depolarization, right? Anytime you're going back down on the graph, we call it repolarization. And if you go down so far that you come back below resting, that would be hyperpolarization, right? Meaning you're extra polarized, you're lower than resting even. And so in the end, this neuron has lots of synapses all over it, right? They're showing the green ones here as being excitatory and the pink ones as being inhibitory. And so it's just a push and pull between, all right, the release of excitatory neurotransmitters gives us EPSPs. We go up on the graph. The release of inhibitory neurotransmitters gives us IPSPs. We go down on the graph, right? Further away from threshold. This value, they're showing it as -55 on the graph. This line, right, is what we call threshold. And if you're technical about it, we really call it threshold voltage because this line is being measured on the y-axis. And the y-axis is membrane potential, which is measured in millivolts. For right now, the way to think about this is threshold is the amount of voltage that you have to reach in order to trigger an action potential. And the action potential on the graph, that's this spike that happens, right? So if you hit this threshold value, you get an action potential. If you don't hit that threshold value, you don't. So EPSPs make an action potential more likely to happen because they send you up towards threshold. And IPSPs make an action potential less likely to happen because they push you down further away from threshold. And this concept of summation is going to come up later even more. But right now, just take it to mean we're combining excitatory and inhibitory synapses that might be firing at about the same time. And we're just averaging them out. So summation to sum, just like in math, if you add positive and negative numbers, they cancel each other out. So if the excitatory signals win, we hit threshold, we get an action potential. If the inhibitory signals win, we don't. Okay. And lastly, just to give you context as to how this graph is being made, this graph isn't just hypothetical. You can take a neuron in a culture dish. So this object that you're seeing here, that is the cell body of a neuron. This would be its axon down here. And then this guy, which looks like a little needle point, that is an electrode, right? That electrode is no different than an electrode that you would plug into, an electrical socket in your house to measure the amount of current that's coming out. You can do this microscopically and measure membrane potential for a cell. So I realized that this graph was kind of a made up, polished example, but in a real lab where neuroscientists are doing work like this, they can put these minor, tiny electrodes inside of any part of a cell they want and measure its membrane potential over time. And they can add excitatory neurotransmitters and inhibitory neurotransmitters and see what happens to the membrane potential of that cell. Okay. All right, so the next part of this, how do neurotransmitters actually cause EPSPs and IPSPs? So in principle, all we really need to know is at a synapse and this picture of a membrane, this membrane would be on the post-synaptic cell. So you could imagine that up here, where it's not being drawn, this would be the axon terminal of the presynaptic cell, right? And that axon terminal is going to contain a bunch of vesicles filled with neurotransmitter. So this particular neurotransmitter that they're using as an example here, ACH, stands for acetylcholine. Those acetylcholine molecules are going to be kept up in these vesicles inside the axon terminal. And when this axon gets an action potential, it's going to release those neurotransmitters into the synapse. Okay, so for context, we're right here, right? The zoom in on that picture on the next slide is showing the release of neurotransmitters by this axon terminal onto the dendrites of the cell body of this postsynaptic neuron. Okay, that's what we're looking at here. And we say that these channels are ligand-gated ion channels, right? So to not get confused, a ligand-gated ion channel and a receptor for the neurotransmitter, in this case, same thing, right? The ion channel is the receptor, and the gated part of it tells you that a gate, just like a gate on a fence can open and close, that there's a gate on this channel that can open and close. And so if we're starting off over here where the neurotransmitter is not bound to the receptor, channel's closed, gate shut, ions can't move through. As soon as the neurotransmitter binds, acetylcholine happens to need two molecules to bind to its receptor. When those acetylcholine neurotransmitter molecules bind to the receptor, the ligand-gated channel, the gate opens, ions can move through, right? And so in this case, they're showing these ions moving from the inside of the cell or the outside of the cell to the inside because that's their gradient, right? These channels are often specific to certain ions. So one ligand-gated ion channel might be specific to letting sodium through. Another one might be specific to calcium or potassium. And so the neurotransmitter is just the signal that we use to decide whether the ions get to flow through or not. Right? And a good set of rules to remember for right now, if you let either sodium or calcium flow through this cell, you are adding a positive charge, right? Sodium and calcium, both positively charged ions, they tend to diffuse into the cell because that's their high to low gradient. They get built up on the outside. They want to flow in. And so sodium and calcium, if they flow into the cell, they tend to cause EPSPs, right? Excitatory, because on your graph, they're going to make you go up and depolarize, getting closer to threshold. If your ligand-gated ion channel is a potassium channel, well, potassium tends to flow out of cells. That would make you lose charge. So you're losing a positive charge. That would make you go more negative on the graph. And so potassium causes IPSPs, inhibitory potentials. So at every individual synapse, you expect to see one particular type of channel. If it's sodium or calcium, it will be excitatory. If it's potassium or chloride for that matter, because chloride ions are negative and they flow in, either of those two will cause IPSPs. And in the end of all this, the question that we're really asking is, were you able to reach threshold? Okay, and so all of these synapses added together, they're firing, some are excitatory, some are inhibitory. All you really care about is, did you have enough excitatory firing relative to the inhibitory to hit that threshold mark? And if you hit threshold, you're going to get this action potential spike. It's an all or nothing response, and it's going to happen as soon as that line is crossed. All right, so before we move on to how the action potential happens, quick clarification. There are actually two different types of receptors. The ones that we looked at in the previous slide, the ligand-gated ion channels, those are the same thing as ionotropic receptors. That's saying what we just talked about. The receptor is the ion channel, and so as soon as the neurotransmitter binds to it, ion channel opens, ion flows through. And so remember, the neurotransmitter itself, that's the red hexagons in this picture, they're not actually making it into the next cell. It's the ions that make it into the cell. And so neurotransmitter binds, channel opens, ions flow through. The other variety of these receptors are called metabotropic receptors because you need some sort of metabolic reaction to have their effect. And so in this case, the end result is the same, but it's indirect. So now our neurotransmitter binds to a receptor. That receptor activates usually a protein called a G protein. And that G protein turns on a signaling cascade that eventually activates an ion channel. And so all that changes is the receptor that the neurotransmitter binds to and the ion channel are two different proteins. They just communicate with each other. But the end result is the same. We open an ion channel. If it's sodium or calcium, it's excitatory. If it's potassium or chloride, it's inhibitory, just direct or indirect. All right. So once we've made that happen, right, our ion channels are opening or closing, the question is, what exactly is it about threshold, right? This exact point that we see on the graph, what is it about reaching threshold that actually makes an ion channel open or close to cause an action potential? And this is where another category comes in. Voltage-gated ion channels are the same concept as ligand-gated, but for ligand-gated ion channels, the binding of the ligand or the neurotransmitter is what makes them open or close. Here, we say that they're voltage-gated because you need a certain amount of voltage to make the channel open. And remember, your threshold line, that's this guy, threshold is measured in millivolts. And so this number, negative 55 millivolts, that's just the amount of voltage or electricity that you need to make this voltage-gated ion channel go from closed to open, right? So anywhere below this threshold line down here, this voltage-gated sodium channel was closed. And as soon as we hit that threshold value, it flings open. And once it opens, just like any other sodium channel, we expect sodium to start entering the cell. Okay, so in this picture, they're showing an ion diffusing the opposite way. But for our case, we care about sodium. So imagine that my black dots are sodium ions. As soon as that voltage-gated channel opens, we have a gradient pointing from high to low. We expect that sodium to diffuse into the cell. Now, the interesting part of this is these voltage-gated channels are only found on axons, right? You don't find them in cell bodies. And so the place where we care about this happening, this red square, that's the beginning of the axon. And if you remember from the intro lecture, that structure is called the axon hillock, which is just the beginning of the axon. And if you hit threshold there, you start a chain reaction. Because what ends up happening is, right, if this is our axon hillock, and we have a lot of these voltage-gated sodium channels inserted in the axon, as soon as you hit threshold back here, right, because you have sodium ions coming in from those synapses, right, the sodium that you released back here at the synapse diffuses to the axon hillock. That's what makes you hit threshold. But as soon as the first voltage-gated sodium channel opens, these red ones that I'm drawing, more sodium is going to enter the cell. That's going to make the next one hit threshold, and it opens. More sodium enters the cell, makes the next one open, makes the next one open. And what you end up getting is this wave of voltage-gated sodium channels opening all the way down this axon until you get to the axon terminal, right? That is an action potential. It's not really any different than someone pressing a button and sending a signal down a wire like an old-fashioned telegram. The electricity pulses down the wire in the same way that this flood of sodium ions coming into the cell pulses down the axon. Okay, and the reason that this is all or none is because it's a chain reaction. It's positive feedback. As soon as you hit that threshold mark and open channel number 1, it's off and running. You know the signal is going to go down the whole cell. It cannot stop. And so once you start an action potential, it's going. And every action potential is the exact same strength, right? It's like flipping a light switch on and off. We either hit threshold or we didn't. And so the shape of this action potential, like how strong it is, like how tall the peak is going to be the same every time. That's why when you measure how action potentials are behaving, the variable that you measure is frequency, which would basically be action potentials per second, right? You can't change how strong each one is, so all you can change is how often you send them, right? And when a neuron wants to send more signals, it just pulses those action potentials faster down the cell. They're like little blips of electrical activity. Okay, so if we're going to get this action potential to travel all the way down the axon, this idea of positive feedback, this is what makes sure that it's all or nothing and it spreads all the way down the axon. The problem is positive feedback also needs some sort of reset mechanism, right? And so the problem is we can't leave those voltage-gated sodium channels open forever, or positive feedback would keep happening indefinitely. And so the solution to that is two things. One is, if you notice in this picture that there's this little ball and chain structure that's blocking the ion channel, when the action potential starts and you hit your peak, that ball and chain very quickly pops back into place to inactivate this channel and make sure that it only stays open for a tiny bit of time. And so our goal in all of this is we want to reset membrane potential and get back down to resting again. So one of the mechanisms is we have to inactivate sodium channels to make sure that they don't stay open for too long. This really only lasts, a fraction of a second. And that inactivation step, right, that prevents this depolarization line from going up and up and up forever, right? So on this graph, -70 is where we started at rest, right? This was our graded potential that we got from releasing neurotransmitters, right? This would be caused by EPSPs. Once you hit threshold right here and we depolarize, the upswing in this line is because during this little time window, those voltage-gated channels are open, right? We hit threshold, they open, sodium is entering the cell at that point. That's why we go up. Once you hit this peak up here, that is where those sodium channels inactivate and cork themselves shut so that you don't keep going up forever. The other thing that happens is axons also have voltage-gated potassium channels. They just open a little bit slower. But when you open a potassium channel, potassium leaves the cell, you're losing a positive ion. That's what actually makes you come back down. Okay. And so the combination of those two things, sodium channels being inactivated and voltage-gated potassium channels being open, they combine to cause repolarization. Think of that as your reset, right? That is getting you back down to resting membrane potential. And very briefly, we even dip below resting membrane potential. That is hyperpolarization, right? Hyper just meaning extra. You're extra polarized because resting membrane potential is right about here. And so for this little time window, you are extra polarized and you go even lower than normal. That's just part of the reset period. And what it leads to is these two things together lead to something called a refractory period. Refractory period really just means a forced reset. We're saying the action potential went up. We want to force it to come all the way back down and get below threshold again. Otherwise, you would risk having the action potential go up and just stay up forever and never reset and come back down. Okay, so there are two parts to this. The absolute refractory period is when sodium channels are inactivated. Okay, that is happening during this time window. When we're repolarizing, that's when inactivation occurs, and it corks those sodium channels shut until you get back below threshold. All right, so during that time, it's absolute. There is no way that you can have another action potential because you have to come back down and reset before the next one happens. Hyperpolarization causes what's called a relative refractory period. And it's relative because at this point, you technically can have another action potential, but because we're below resting, it's relatively more difficult. Okay, so if you wanted to get another action potential here, this would be possible, but you would need very strong EPSPs that would get you back up to threshold, and then you could get another spike, right? But because we're below resting, there's further to go to get from here to threshold than there was from resting to threshold. And so it's relatively more difficult. You need a stronger stimulus to make it happen. Once we've recovered, all the potassium channels are shut. Because those potassium channels being open, that's what's causing hyperpolarization. Once they're all closed and we're back to true resting, then we're back to normal. The refractory period's over. And that's exactly the same as where we started. Okay. So I hope this was a good summary. And follow along in the playlist. The resting membrane potential video before this is critical to understanding this. And the one that's going to come after, where we talk about myelin and ions diffusing down axons and eventually synapses, these all go together in a series. So flag the playlist, subscribe to the videos so that you can put them all together and understand the entire topic.